Just a moment. Yeah, so I am Teemu Daniel Laajala. I'm here today to represent our team FIM U2, which comes from the collaboration between the Institute for Molecular Medicine Finland, University of Helsinki, and Department of Mathematics and Statistics in University of Turku. And like Jim already presented, I'm going to be focusing on the sub-challenge 1A for obvious reasons, and the topic of my talk is predicting the patient's survival in the DREAM 9.5 metastatic castration-resistant prostate cancer challenge. So without further ado, I'm actually going to start with showing the very beginning and the very end of what we did. So over in... Is there a laser pointer here? Yeah. So over here in the top left corner, we actually have an extract of our kickoff memo that was done in April 15th, 2015. We predicted 3 key factors to be determining for a winning team in this sub-challenge 1A that I will be focusing on. Point number 1, we thought that based on our preliminary research, exploratory research on the data, we saw that there would be quite big study-wise effects. So the collection of data between the different cohorts, some other sources of variation. So we thought that that's one of the main criteria. Second one, as anybody who's done survival analysis, it's a special type of analysis where right censoring occurs. Either you observe a death or you only know for how long somebody survived. So we predicted that the winning team would somehow utilize a model family that is tailored for this purpose. For example, survivor forests or Cox regression or so forth. And we actually went with Cox regression later on. And third, we predicted that the ability to adjust or impute missingness in the data, especially the structured. I will be going further into that into detail, but there was some study-wise structured missingness there. And then what we, in the end, what we did, I'll split it into 4 categories. So there's the pre-processing of the data, like Liz already presented. There's the really neat core table, but we also used other sources. And I think that's an important mark there. I will go into detail into that, but then we proceeded into unsupervised learning. So we're looking at the distributional properties of the data, consulting a clinical expert, and so forth. And then we moved on to supervised learning, and we actually went with ensemble learning. So I will tell more about that, and obviously in the end we then did ensemble predictions and outperformed the other competing teams. So a word about the data pre-processing and imputation. This is actually a missingness matrix of the data itself. In columns we have the variables, and in rows we have the patients. So we had a large chunk in the data which was present, but we also observed really big chunks of structured missingness in the variables that we extracted, somewhere from the core table, somewhere from lab values and vital signs. We didn't include the other longitudinal tables. And we split this missingness into 2 main categories: missing at random, which might not be entirely true, but we put a threshold that maximum 2% of values missing for a certain variable, it would be treated as missing at random. And then structured missingness, which was very much connected to each of the 4 studies where AstraZeneca was the target study to be predicted. So what we actually did was Gaussian penalized regression. And we started off with the full matrix over here. So these variables were fully observed, and we then filled in the missing at random gaps. So what we did was take all the observed values for a variable at a time, teach the model based on cross-validation on this fully observed data, and then predict the missing pieces, the black dots. So that was our missing at random part. But then came the structured missingness. We utilized a similar model, but then we kind of accumulated more data by also including the MAR imputed data matrix X, which allowed us to have much more variables into predicting these large chunks that you see in the sides, especially. The nice thing about this Gaussian penalized regression is that since we're doing cross-validation and we're fitting a model to impute rather than use some naive thing, we actually are trying to test if there is informativeness here in the variables to fill in the gaps. And if there is no informativeness, the only term that will be left is the intercept term in such linear regression model, and that actually will then correspond to mean imputation, which is kind of a naive benchmark, if you will. So it has a nice connection. So there's a possibility for a model if we find informative variables, but if not, then we will resort to mean imputation. Then moving on to unsupervised learning. We did take a look at the distributional characteristics of each of the 4 studies, and since it's a competition and the AstraZeneca study was the target set, we did have to do some research, especially with that AstraZeneca in mind. And here I have a very important example of bone metastasis, bone lesions from the core tables. And what we observed was that in the training part of the data, we had a really nice distribution, more of the positive cases, but some negative cases. But in the target set, we would have everything as positive. So there would be no predictive information available for the model in the target set. So when we're training our data based on this, it would probably pick up this variable, but we would suffer in the sense that when we're then doing the predictions for AstraZeneca, we will not have that information available. For some reason, all the patients there were marked as metastatic in bone. So this was one reason that we, for example, dropped out this covariate from our analysis. I then, in our team, we had a clinical expert, Tuomas Mirti, prostate cancer expert, and I interviewed him about the covariates and whether they would be relevant or not, and he would indicate to me whether some, for example, TERP, which means cutting off some extra cells due to hyperplasia, if we assume that it meant for that, then it would not be relevant and we could discard that. Gastroreflux disease, not relevant. Medical history of eye disease is not relevant. So we did some kind of retrospective dropping out of non-relevant features. But also then there were obviously a lot of maybes involved, for example, spinal cord surgery. So we thought that if it has been done due to metastasis, then it's relevant. But we also observed the distributional properties, and in the end, we, for example, discarded this variable because it didn't have enough informative power, in our opinion. And then an example, positive example of a variable that we kept, it was annotated as very important, so we kept it along. We proceeded with unsupervised learning, and I'm assuming that most of you are familiar with principal component analysis. So we took a look at this fully observable data matrix that we had created, and over here we have the mean of the target set, AstraZeneca, and then we have a cell gene over here and mean EFC here. And we already observed here that EFC, on the left-hand side, we had all the variables that we had available at baseline when we were studying the supervised process. And we already observed that most likely cell gene and EFC are going to be most relevant. They are closest in the unsupervised learning to the AstraZeneca set. And then we took a closer look to the data, and we observed that if we took only the binary variables, we could see a clear distinction to the ASCENT-2 study. And this kind of raised the suspicion on whether there is something that is not representative of the AstraZeneca study, which is actually its mean for the binary variables is over here, and EFC here, and cell gene here. So we did try to find what parts of the data are most relevant for the question in mind. Here's an interesting plot when we were transitioning from unsupervised to supervised learning. On the y-axis, we have the first principal component, so the component corresponding to most of the variation in the data. But on the x-axis, we have the response vector y. And in here, we have a follow-up in this axis. Oh. Okay, I'm back. There were obvious differences between the follow-up times in the studies. For example, this EFC study, which is in green, each green dot is a patient, and we know the patient's status in the training set. So we knew which patients died at what time, and then we had the right-censored patients. And AstraZeneca is shown in the negative axis because we didn't know the correct answer to the y vector. This was the obvious, it was the whole purpose of the competition to predict this AstraZeneca. But what we observed also here is that ASCEN2 was kind of accumulating to a certain area in the principal component. While on the other hand, Celgene and EFC would have a rather nice coverage on the y-axis, which was the first principal component. So it would cover most of the variance that we would observe in AstraZeneca. Another thing that we had in mind is that we wanted to have the response vector properties as similar to AstraZeneca as possible. And what we observed then, it was kind of a win-win situation in the sense that we observed that Celgene and EFC are also if you take the mean of their the organizers gave out the median follow-up time for AstraZeneca study, so it was 463 days. We didn't know anything else about the minimum and maximum, but the median was most important. And what we actually observed is that if we take the Celgene study and the EFC study, the mean of those two follow-ups is actually pretty close to what would be the properties of the AstraZeneca study. So we were at this point a little bit suspicious whether we would, what we would do with the ASCEN study. So here's a glimpse on the response vector Y and the data matrix X. So as per survival analysis, you have the death status, 0 meaning right censoring, we haven't observed death, and 1 meaning death, and then a certain amount of days. And then this is approximately how our data matrix would look like. We would have log transformed variables, for example, due to making the distributional properties better in those variables. And then we would have quite many binary variables, for example, opioid analgesics use, which was part of the Halabi model. That's why I'm including it here. But what we went then to do is we introduced some nonlinearity into the model by introducing pairwise interaction terms. For example, taking the pairwise interaction between these two variables. In the beginning, when we started, we had 124 variables. Like I mentioned, we had variables beyond the core table, and they would actually be very valuable in the end. After doing the unsupervised learning, looking into the distributional properties and consulting our clinical expert, we ended up with 101 variables that would be interesting. And then again, we started expanding the data matrix by introducing those nonlinear components, not all possible pairwise components, but we ended up with over 3,000 variables in our model, or well, in the data matrix. And those of you that do machine learning, you might be familiar with this particular formula. We went for penalized Cox regression. So on the left-hand side, you have the model likelihood. And when the model is fit, we basically do embedded feature selection. So there's the penalization coefficient lambda in the term that tries to balance out model likelihood versus how much the model coefficients beta deviate from 0. So this lambda will be estimated using cross-validation to find a good weighting of likelihood versus non-zero coefficients. And then we have the parameter alpha. And actually, the Halabi model was a lasso model, so it has a alpha value of 1. So basically, one of the terms disappearing from here. But then we wanted to explore more on that. So we wanted to test different values of alpha, and so we tested a whole range of elastic net models and also ridge regression, which is a particular special case of this formula when alpha is 0. And since it was survival analysis type of data, it was important that we would utilize Cox regression, and it could be used for predicting hazard ratios. What we observed in our cross-validation, we observed that the study-wise eAUCs were very sensitive to what studies we would include. And so we decided to test different ensemble components, different models, and collect them together. And so we could better optimize the eAUC on those components. And those we were hoping that those would better account for the study-specific characteristics. And at this point, we actually did the drastic decision that we omitted ascent 2 from our final model. And we ended up with an ensemble model, or ensemble of 3 models. One that was trained on cell gene only, one that was trained on EFC only, and a model that was trained on their combination. And then these 3 components would be summarized into an ensemble prediction by taking the average of the risks per prediction in each component. And since the prediction is like kind of an arbitrary ratio of risks, we took the average of ranks rather than average of the predicted values themselves. Here's what the optimization space looked like. We were simultaneously optimizing alpha to find suitable lasso versus ridge regression and elastic net on the y-axis here, and then suitable lambda for the penalization coefficient. So we actually found out that pretty many of the components that we could think of, we iterated all different combinations. Most of them were actually very close to ridge regression, which might be partly because we had so many coefficients in our data matrix, over 3,000. But there was one exception, which was the ascent plus cell gene. If we combined those two, that was actually somewhat lassoic elastic net alpha value 0.78. But otherwise, we would be pretty much dealing with ridge regression, very low alpha values. So that would mean that a lot of coefficients would be conserved in the model. And lasso has the nice property, on the other hand, that it selects very few. So, but this was based on the cross-validation where we observed the optimal value. So that's why we did the ridge regression type. Then after the competition and giving out our final ensemble prediction, we wanted to find out, so what were some of the key factors then in the coefficients that really did contribute to the winning model. And we actually found out that many of blood and liver related markers, and for example, hematocrit, which was extracted from one of the tables manually, was one of the top markers. It wasn't included in the table by, I believe, Jim, because it was not included in the core table, if I recall right. Then some other more familiar names like aspartate aminotransferase and red blood cell count, and one binary indicator if the cancer lesions were found in liver. And we also did identify quite many of the Halabi model markers, so I'm underlining the Halabi model markers. If we took the absolute effect on the hazard ratio averaged over the ensemble components at the optimal point in the alpha lambda space, we did get very different coefficients than in, this is just top 10. And we did observe, for example, the lactate dehydrogenase. But if we look at the shrinkage ranks, so looking at this figure, so when you increase lambda, you push more and more of the coefficients towards 0. So we computed the rank at what point each coefficient would be shrunk exactly to 0. And there we had more of the Halabi type of variables, for example, LDH, the alkaline phosphatase, hemoglobin, and such. But even there, we got some hematocrit and specific gravity related to urine and such. And what we actually think is that a lot of bone healthiness and liver-related markers were popping up. And these are both post-competition validations that we're doing. This validation was done internally in the sense that we didn't show AstraZeneca data to it at all, just the trained model and observe the coefficients and try to infer which are most important. But then we did also external validation in the sense that we submitted new submissions to the system as allowed by the organizers to check what factors really influenced our model in regards to predicting AstraZeneca well. So, for example, we tried omitting a variable and all possible interactions of it one at a time. And the top 5 hits were eosinophiles, which I actually, I'm not an immunologist, but I believe there are white blood cells that mature in the bone marrow. So they're related to bone. So that if we omitted that variable, it actually reduced the AUC the most. So that was the most important variable in our model. 2 of the Halabi markers were among the top 5, but also liver was 5th best in this respect. But we also observed that if we omitted some coefficients along with the interactions, we would improve the model. And it was actually interesting that opioid analgesic use was the second kind of confounding variable in our model. So if we omitted that, we would get an improvement to our AUC. Then we also tried to figure out whether the ensemble combination, how we combine these different structures, if we could improve that. But we actually got the conclusion that we pretty much hit the gold spot. We had the best possible composition. We tried different models, but some that also included Ascent came pretty close. You can't see it because of the side, but almost some of them were as good as the one that we finally submitted. But in the end, the best performing one didn't include Ascent 2. As a summary, we utilized both unsupervised learning and clinical expertise to improve our performance. Our optimization of alpha and lambda favored ridge regression-like models. We used ensemble learning to account for the study-wise effects. The role of the Ascent 2 study, from our perspective, remains a little bit unclear how we should incorporate that now. But the cell gene and EFC were most important for predicting AZ, AstraZeneca target set. We believe that bone and liver healthiness or markers related to those were the most important ones, both based on the internal and external validation, meaning without and with AstraZeneca information available. So without further ado, that's our team, and thank you for your time, and now hopefully we still have time for questions. And please also visit me at the poster if you got further questions, if we're running out of time here.
So we do have time for just one question. I'm gonna switch presentations.
I have a very quick question regarding your regression. Could it be the reason that you favor ridge regression dependency among variables that make the problem ill-conditioned? So you had to use ridge regression to make sure eigenvalues are further from 0. So that would be my first question. And in that case, would it make sense to do pre-processing and minimize those dependencies before actually using regression?
Well, from our perspective, using ridge regression in terms of the accuracy wasn't a problem. So we didn't really want to avoid ridge regression, even though usually people want to use lasso or at least close to lasso because it selects fewer variables. But we did see one component that was very high in alpha. So I'm not sure if it was about the data structure and such, because depending on which studies we combine to the optimization procedure, we could also get a model that had relatively high alpha if we maximized the AUC in tenfold cross-validation. So it's hard to say whether that would improve or if we should really avoid ridge regression-like, but it would certainly be easier to infer the models if they weren't ridge regression-like.
Oh, so I'm just wondering, how well do these variables predict survival in normal people? So if you had normal people but with the same variables, so as to know whether the survival you're predicting is not related to disease.
Yeah, that is of course very interesting, and for this purpose we did collaborate a lot with our team's clinician Tuomas Mirti, and we did try to find a biological insight into why these markers were as good as they were. But in order to have a correct answer to your question in a normal population, I think we should have such a data set to test it out, and it's really hard to say straightforward without having that type of data. But we did kind of also keep the biological inference in mind, so the markers that we found do have some explanation to them.
Oh,
so
okay. I was just wondering because perhaps that would be the random model, like the effect of those variables on the normal population as a random or background model.
Yeah.
One more.
How much worse is the performance if you just do mean imputation rather than the model-based imputation?
That's a very good question. We actually explored quite a few different methods.
Repeat the question.
So the question was that how much better did we perform just because we did model-based imputation instead of, for example, mean imputation or median imputation, which could be kind of seen as more naive approaches. What I actually did in the beginning, since I'm only presenting the end results now, is that we tried cross-validation, for example, with KNN imputation, K nearest neighbors, and we did see some effect in the metric, and we did see, for example, when we were cross-validating that. I didn't actually do systematic testing for this anymore because I had already observed that the imputation technique would affect the end metric. But what I particularly liked here is that since we're building a model, the cross-validation itself should handle the fact that if there is no informativeness, then it will go to mean imputation because it will shrink all the variables and leave out the intercept. So it does have an effect to, I don't think it was, it's hard to pinpoint just a single factor that would have led to this since we were statistically significantly best team, but it does have an effect. So you might want to consider, especially given that there was this structured missingness. So like I tried to underline that, it's really hard to account for.